Welcome to InVitro's webinar series. Today we have Drs. Kenneth Bloom and Matthew Silva who will address in vivo and tissue molecular imaging and gene therapy. Dr. Bloom is Chief Medical Officer of Advanced Genomics and Pathology Services for Konica Minolta's precision medicines businesses InVitro and Ambry Genetics. Dr. Bloom has more than 30 years of clinical experience and has been an early adopter of precision medicine. Prior to joining InVitro, Dr. Bloom was president and head of oncology and immunotherapy for Human Longevity Incorporated, and prior to served as chief medical officer of in vitro diagnostics life sciences at GE. In addition, Dr. Bloom served as president and CEO of the medical group of Clariant Pathology Services. Dr. Bloom received his medical degree from Rush Medical College. Dr. Matthew Silva is InVitro's EVP of Scientific Applications and brings 16 years of diverse imaging experience from pharmaceutical and contract research organizations. Prior to InVitro, Dr. Silva was Senior Director of Translational Biology at Vertex Pharmaceuticals, where he ran the in vivo imaging and histopathology groups. Dr. Silva also previously held leadership roles at Amgen, Millennium, and Takeda Pharmaceuticals. Dr. Silva has a PhD and master's in biomedical engineering from Worcester Polytechnic Institute. We are pleased to have Dr. Bloom and Dr. Silva with us today. Before we get started, attendees, please note you may ask questions via the Q&A box throughout the presentation. At the end of the presentation, the panelists will address them accordingly. Welcome, Dr. Bloom and Dr. Silva.
Thank you, everybody, for attending today's webinar on advanced biomarker tools to support gene therapy research. Matt and I will be tag teaming this event, covering key challenges, emerging techniques, tools, and a few case studies spanning preclinical and clinical research. There are several challenges associated with gene therapy, including patient selection, pharmacokinetics, integration, expression, pharmacodynamics, and safety. Several questions need to be addressed. How will the correct patients be identified for a specific gene therapy? Is there a stratification scheme based on mutational analysis? Are there advantages to using one genomic method over others? There are also questions specifically about the therapy itself. Does my therapy reach the target tissue? What about off-target uptake? What is the half-life in blood? What is the half-life in the target tissue? And are they even related? What's the optimal route of administration, the optimal dose, and schedule? Does my DNA integrate properly? And what is the integration efficiency? Do I see the expected expression? What is the expression efficiency? What is its duration and stability? Is the expression uniform in the target organ? What about expression in non-target tissue? If expression is confirmed, is there a biomarker of response? Is there a relationship between uptake, expression, and distribution? And of course, there are always safety concerns. Is there therapy itself immunogenic? Is there off-target or non-target modifications? And are there long-term consequences? So what about the methods that are in use? Patient selection methods are usually targeted, but are moving more broad, like whole exome or whole genome sequencing. Pharmacokinetics have traditionally been assessed as tissue homogenates by liquid chromatography, mass spectroscopy, immunoblotting, PCR, and more recently by immunohistochemistry. Integration is typically assessed by cell-based assays. Current methods to assess pharmacoexpression include immunohistochemistry, immunofluorescence, and flow cytometry. In vivo methods include bioluminescent and fluorescent imaging or nuclear medicine imaging, such as SPECT or PET, and are frequently used via reporter genes. Pharmacodynamic markers typically include tissue-based immunohistochemistry, blood-based biomarkers, or imaging methods. Safety is assessed by ELISA or other techniques looking for anti-drug antibodies, viral integration assessment, and other custom safety panels. We will be talking about emerging methods in gene therapy, including whole genome sequencing and RNA-seq for patient selection, quantitative whole body imaging of AAV and ASO distribution, in vivo and ex vivo whole body imaging of expression, and quantitative whole body imaging of treatment response. We do in vivo and ex vivo imaging spanning preclinical discovery through clinical assessment utilizing micro to macro resolution methods. The image on the left demonstrates a size 7 labeled antisense oligonucleotide following intrathecal administration. The brain, the excised brain, was imaged using a technique that I will explain a little bit later called cryofluorescence tomography to determine whole brain distribution on the macro level. A mid-level section was collected and stained for several cellular markers to image and co-localize the antisense oligonucleotide on a cellular level. In this example, we were able to confirm that the antisense oligonucleotide crossed the blood-brain barrier and reached the neuronal nuclei. The next image over demonstrates a similarly radio-labeled antisense oligonucleotide that was imaged by SPECT CT to determine the pharmacokinetics. The imaging highlights the neuroaxis with renal and lymphatic clearance. The next image over demonstrates the AAV-mediated GFP expression in a whole mouse using cryofluorescence tomography to provide high-resolution 3D images. And the image on the far right demonstrates a radio-labeled ASO in a human following intrathecal administration to determine biodistribution, safety, and dosimetry. The complete spectrum of imaging techniques are necessary to follow pharmacokinetics, pharmacoexpression, and pharmacodynamics of gene therapy. Imaging is essential to determine the fate of the vector. The distribution must be tracked accurately over time. The transduction and consequent expression of the delivered gene must be confirmed, and the therapeutic consequences must be evaluated. Ex vivo and in vivo imaging techniques have been developed to monitor the pharmacokinetics of the drug product, evaluate whole body expression, and confirm biologic activity. Transporters such as membrane receptors enable cellular internalization of probes in target cells, and products are visualized with the use of enzymes which can phosphorylate reporter genes that get trapped in target cells. This slide depicts the traditional methods of determining the distribution and expression of an adeno-associated virus vector, or AAV. On the left side, we see data from a study in which distribution and expression of the AAV vector was determined by sampling a variety of different organs for expression by PCR. The tissue from each organ is typically homogenized and then evaluated, so it is impossible to determine regional differences of expression within the organ. On the right side, we see tissue sections from different regions of the spinal cord 3 weeks after delivery of an AAV9 vector. Green fluorescent protein expression can be seen in the dorsal root ganglion neurons in the animals which were injected intrathecally, but not in the animals that were injected intravenously. We believe that we can improve on these techniques. We utilize cryofluorescent tomography to provide high-resolution 3D images across whole animals, significantly improving the assessment of distribution and expression. This modality is extremely useful to image vector-mediated expression of fluorescent proteins and can be beneficial in screening gene delivery vehicles. In this slide, we demonstrate AAV9-mediated green fluorescent protein pharmacoexpression in a whole mouse. Our typical output includes fly-through of both the bright field and the fluorescent 2D images as they are acquired from the block face, as well as 3D reconstruction images. Positive signal is detected in several organs, in this example, including the liver, heart, and dorsal root ganglia. Control images show faint autofluorescence from the gastrointestinal tract. Cryofluorescent tomography is an ex vivo multimodality imaging technique that provides white light anatomical and molecular fluorescent 3D images with micron scale resolution. CFT bridges the gap between fluorescence histology and bulk fluorescence imaging by providing high resolution imaging in a range of sample sizes. In this technique, the block-based 2D bright field and fluorescence images are captured using sequential serial sections, as you can see in the cartoon, or in the animation. Histologic sections can then be selected based on real-time molecular information, eliminating the guesswork in collection. 3D mapping of the fluorescent signal is done with anatomical white light reference using in vitro software platform. Images can be viewed in a variety of formats, including the fly-through example shown previously. Imaging allows us to tackle complex questions, like what is the biodistribution of the gene-carrying vehicle, what is the fate of the vehicle, and where is the payload delivered? On the left, you can see the biodistribution of a radio-labeled AAV in a rat. SPECT-CT was performed at several time points, demonstrating the biodistribution along the neuroaxis. The image shown is 1 hour post-intrathecal administration. 4 weeks following intrathecal administration, the rats were sacrificed and imaged whole to assess the distribution of the infrared fluorescent protein, which was delivered as a gene by the AAVs. We can see a discrepancy between the distribution and the transduction of the AAVs. On the far right, SPECT, CT, and cryofluorescent tomography images are merged through in vitro software to better visualize the data. To further localize the expression of infrared fluorescent protein along the neuroaxis, we collected a section for histologic evaluation from the cryofluorescence tomography process. The section was stained and imaged under a microscope in high resolution. We were able to confirm that the signal detected along the vertebra is indeed localized in muscle surrounding the vertebra and may be the result of AAV leakage through the intrathecal catheter installed. Next slide. Sometimes the expression might be quite weak, requiring a high sensitivity technique for visualization. Konica Minolta has developed fluorescent nanoparticles called phosphor integrated dots, or PIDs, that display features that solve many of the inherent problems with quantitative immunohistochemistry and significantly increases the sensitivity of traditional immunohistochemistry without the need for amplification. We know that fluorescent labels increase the sensitivity of IHC relative to chromogenic labels and may be quantitative, since the intensity of the fluorescence is proportional to the intensity of the photon excitation energy. The fluorescence intensity of PID particles is roughly 30,000-fold greater than a standard fluorescent signal, and thus is not hindered by tissue autofluorescence. Moreover, a single PID has approximately 2,500 strep-avidin molecules on its surface, and thus exhibits high reactivity to secondary antibodies. PIDs can be applied to quantify target proteins, antibody drugs, or antibody drug conjugates. The bright spots resulting from PID staining have been simultaneously imaged using fluorescence microscopy and scanning electron microscopy, as depicted in the center image. The patterns resulting from hundreds of positive bright spots were analyzed, and data from the fluorescent signals and the particle number determined by electron microscopy, a standard curve was obtained and can now be used to determine the number of PID particles per bright spot. Next slide. PIDs can be used as an enhanced detection system for most immunohistochemical antibodies. This slide depicts a section of rat brain in which the expression of an AAV-mediated protein has been visualized with PIDs. The results are quantitative and can be reported as expression per cell or per area, as in this example. Our combination of cryofluorescence tomography coupled with PID technology gives us the ability to assess AAV-mediated expression and distribution at a level far beyond what has been traditionally available. And now I'll pass the baton over to Matt, who will continue the presentation.
Thank you very much, Ken. So Ken described fantastically some of the ex vivo techniques that we use to evaluate gene products or gene delivery methodologies. And now we'll move more towards the in vivo assessment. What are the correlative or complementary in vivo tools that we can use to confirm pharmacokinetics, expression, and dynamics? I'd like to begin first with kinetics and some work that was done earlier to evaluate delivery of agents like AAVs. In this particular example, the team used a formulation of AAV that was infused with iron oxides that could be used by MRI to provide dark contrast in the locations of where the injectate went. These were fantastic early implementations, confirmation of accurate injection site. However, this is not a surrogate for the PK of the AAV itself. Rather, it shows you what the location of the iron oxide is and not the AAV. So some of the work that we have been performing has really been to address that fundamental problem, evaluating the AAV or the ASO or whatever the gene therapy product is directly. Switching gears a little bit, I want to talk about therapeutic antisense oligonucleotides. Like AAVs, they're an effective strategy in gene therapy. However, they suffer from the fact that if the target organ is the brain, they do not cross the blood-brain barrier. And as Ken alluded to, what we've been doing is direct administration into the intrathecal space to overcome this limitation. As many know, the ASO works by blocking mRNA and preventing translation and production of a protein. But the requirement to bypass the BBB to get to the brain requires administration via the intrathecal or subarachnoid space by a spinal cord, often called a tap, when you're going to be taking fluids. In this case, it's rather the administration of a fluid. As Ken showed, we've got a variety of techniques that we can utilize to confirm that we get delivery to the site. And the ex vivo tools provide exquisite sensitivity. However, they do not allow you to look at different time points very effectively. So we started to develop tools, many in collaboration with Dr. AJ Verma, formerly of Biogen, who'll be detailing more of this intrathecal work at an upcoming talk in Boston in April, but really began to think carefully about how injection rate, volume, formulation, molecular weight, charge, all of those aspects of the drug itself, how they can impact the intrathecal delivery route, and to develop preclinical tools that would allow us to assess this. We began that work very simply by looking at something like gadolinium DTPA kinetics in a rat, and there's a couple of example movies playing on the right showing how that enhancement changes over time. In this case, DTPA is not reacting, it's not targeted, there is no target for this compound, so it's just really a measure of distribution and then clearance. But what we were setting up for was work to support ASO delivery and expression studies that were done in collaboration with the team at Biogen, Ionis, as well as folks here at InVicro. And I'll walk through an example of some work that was presented at SFN in 2018, in which there was a control group using an ASO against MALAT1, the non-coding region, and then also an ASO against GABRA1, which is a subunit, a critical subunit in the GABA-A receptor. And to want to show how we established the biodistribution of the ASO the expression of it, and then subsequently the pharmacodynamic effect of actually blocking that GABA subunit production. Some of the initial work that we did after performing the MRI was to look at the intrathecal route as a function of the type of molecule. So in an early proof of concept study, using the same DTPA that would be used for MRI, rather than using a metal that is visible by MRI, we switched to a radio metal, which is visible by SPECT imaging. So that's indium-111 DTPA, and compared that to a routinely used clinical agent called technetium-99 sestamib, or shorthand MIBI. MIBI scans are used to look at myocardial perfusion. In this case, we were using it just as an agent that would have some potential for uptake in cells along the spinal column after injection. As you can see in the image on the left, 0 to 15 minutes after the intrathecal bolus, both signals are in the area of the injection, more caudal in the animal. After about 2 hours, there is a visual separation between the sestamib signal, which is in more of the greenish tones, as compared to the DTPA, which is in the blues and reds. And at 6 hours, there's a more stark difference. While the images are useful, the quantification shown on the right-hand side is more illustrative of the effect. And just to point out a single curve here, looking at 6 hours, the DTPA has extended the entire way through the neural axis from the injection site and up into the brain, whereas the sestamib has not, indicating that there's local binding or something that is otherwise preventing the distribution to the brain. This is a critical factor. Although these are test examples and neither of them are gene therapy products, it really is an example of how the product that's being administered can have a differential PK and distribution. This work was extended logically to test the ASO and what the distribution of the ASO would be. I think one of the most important factors here that's really being downplayed is the amount of work that it took to stably radio label the ASO to ensure that it is not being metabolized or dehalogenated in the body, losing the iodine, which would then just clear and be not useful. But following confirmation that we could stably label the ASO, you can see in this example, a Sprague Dawley rat injected intrathecally with a couple hundred microcuries of the I-125 material. I-125 has a 60-day half-life and is visible by SPECT imaging, so it's a good agent for us to evaluate long distribution, especially since we didn't know what the kinetics were going to be specifically, not just the kinetics of getting to the brain, but as you can see, beginning 1, 2, and 7 days after administration, we're also observing clearance. In this particular example, we were routinely administering about 30 microliters, followed by a 40 microliter flush to assist the injectate in getting up into the brain. In panel A, you can see the whole body images, and the image all the way on the right is a confirmatory autoradiography image that was acquired at the end of the study. And in panels B, you can see just a zoom in of the brain where you can see that there is a differential uptake into the brain and then subsequent clearance from the brain over time. Ken introduced the CFT, the cryofluorescence tomography method. This was also deployed in this particular study. While the in vivo imaging is highly valuable to look at both early and late time points and to better understand kinetics, it does lack the spatial resolution that CFT offers. And so CFT was also deployed in this study. As you can see, really just focusing on the center panel where you're looking at sections through the animal brain, 1 hour, 2, 4, 6, 8, and 12 after administration using a very similar protocol before. The difference here is rather than a radio-labeled version of the ASO, we're looking at a Psi-7 labeled version of the ASO. and then subjecting those brains to CFT, you're able to visualize the distribution of that fluorescent agent from the parenchymal spaces and into the brain very effectively over time. On the right-hand side, these two-dimensional images can be rendered fully in 3 dimensions, so the spatial visualization is very clear. The next point of the study was we wanted to evaluate whether or not there was a pharmacodynamic effect. So we've been able to visualize that the ASO was to distribute to the brain, but then the really critical question is it functionally active? Is it performing the function that we intend it to? So the GABA1 gene, as I mentioned earlier, encodes for the alpha-1 subunit of the GABA-A receptor protein. And in this case, this was a fantastic and very purposefully chosen example. There is an existing PET ligand for the GABA receptor called flumazenil. This is a clinically used tracer that can be used to evaluate GABA receptor or drugs that are targeting GABA specifically. In this case, we were administering the control, the mallet-1, as well as the GABA1 specific ASO. When the mallet-1 was injected intrathecally, and subsequently after a few weeks, the binding and imaging of flumazenil is normal, as indicated in that top panel. That's the anticipated binding in a normal rat of flumazenil, representing the GABA-A receptor density. However, when the GABA1 ASO is injected and then flumazenil PET is performed after a few weeks, you can see in the bottom row of images a reduction in binding of that PET ligand, indicating that there is, the ASO is in fact blocking production of that alpha-1 subunit and preventing formation of the GABA-A receptor effectively. In this case, this was a well-chosen example because there was the existing PET ligand to perform the work. However, it is very feasible for novel targets that a custom PET ligand be developed to look at proteins or receptors of interest. Looking at that data a little bit closer, we used a brain atlas to look at specific regions, to look at regional changes. The images on the right-hand side are really the time activity curves showing you the uptake as a function of time and over 4 weeks. This data is summarized on the right-hand side where you can see the area under the curve relative to baseline for flumazenil binding. The red curve is the GABRA, where we anticipate there is going to be a reduction in the GABA binding, and that is indicated here. You can see nearly a 50% reduction in cortex, the cerebellum, brainstem, and striatum, whereas in the mallet-1, there is statistically no change in flumazenil over time. In the same study, what is really impressive about these techniques is that we can couple the in vivo and the ex vivo and several methodologies to very intimately understand the distribution, expression, and kinetics. So in this case, you're looking at here the effect of the expression of GABA1 by immunohistochemistry. The mallet-1, there is a normal expression. In mallet-1, there's a normal expression of GABA, whereas in the GABRA1, there is a reduction, as indicated in the region that has been highlighted, the cerebellum. In panel C, you can see there's a reduction in RNA. There's a reduction in IHC. And these are confirming what we saw in the in vivo PET imaging, where there's a reduction in the flumazenil AUC. So across in vivo and ex vivo methods, we can see confirmation of an effect. For clinical translation, however, iodine-125 is not suitable. It is a fantastic research tool. It allows us to ask a lot of questions in animal models, and it can prepare us for clinical trials, but it in and of itself isn't a suitable radiotracer for the clinic. To overcome that, the team spent a lot of time trying to adapt those methodologies for a clinically translatable tool. In this case, using technetium-99 and a mag3 chelator that is coupled to the ASO. A couple of years of work went into optimizing this methodology to ensure that it would be stable and suitable, and to bring this up to GMP production standards. Before considering a clinical trial, it is often very useful to look in a larger species. Here in non-human primates, we're able to demonstrate that intrathecally administered Tec-99 mag3 ASO enables us to obtain high-quality images. Here you're looking at a single non-human primate with images acquired at several time points up into 24 hours. At 24 hours, you're beginning to see more prominently the kidneys. So again, we're understanding not just that we're able to access the drug site of interest, but we're also able to confirm clearance. Following that, and I do not yet have human ASO data to show you, but rather it was important first to develop methods to look at dosimetry and do proof of concept intrathecal administration in humans. These are some of those proof of concept scans published by Hesterman et al., in which they evaluated the Tec-99 DTPA administered intrathecally in human subjects. What was critically important in this particular work is to assess the safety, the radiodosimetry and safety associated with injecting a radioactive tracer into the intrathecal space. These models had not yet been developed, but the team here, in collaboration with others, were able to perform this first imaging, as well as develop the models for dosimetry associated with it. So all in all, I think what I hope that this shows is a methodology that takes us from the early preclinical experiments and into the clinic that allow us to look at ex vivo tissues as well as in vivo images to understand kinetics, distribution, and pharmacodynamics. With that, I'll turn it back over to Ken to finish off as it pertains to patient selection.
Thanks, Matt. So one of the new capabilities that we have as in vitro is that we're a member of the Konica Benilta Precision Medicine family. And one of our sister companies, Ambry Genetics, is one of the world's leader in assessing germline genetics. So patient selection requires an accurate and rapid genetic testing. And through Ambry Genetics, we have this capability to run either single gene tests, multi-gene panels, or even whole exome or whole genome sequencing. The purpose of selecting the patients through genomic analysis is to attempt to identify patients that have the targeted mutation and pull them out from patients without the targeted mutation. To do that, you really need a company that has an advanced team focused on patient identification. And through Ambry, we've been able to create that through better field team execution, more flexibility in arranging our panels to attract leading KOLs. And most importantly, one of the big differentiators of Ambry Genetics is the ability to better resolve variants of unknown significance into likely pathogenic or likely benign. Next slide. And so this is summarized on the next slide. Again, the pharma need is generally to identify patients many times with seemingly unrelated symptoms in multiple organs being seen by numerous doctors across multiple specialties. So it's not always obvious who has that mutational defect. And of course, pharma wants to overcome the competitive commercial challenges that go into identifying rare patients with disease. So Ambry's solution has been to tailor genetic testing panels specifically to individual needs to maximize patient identification. The ability to customize the test menu allows the client to select the genes of interest rather than running broad, expensive panels that could create confusion in patient selection. broad field support, including over 100 genetic counselors on call to aid physicians in answering questions about molecular testing, which could sometimes lead to some very interesting questions. And of course, we've done this many times in the past, so the ability to integrate the Ambry sales team with pharma sales team becomes critical. And as you know, when you do genetic testing, you really do need genetic counselors to provide pre and post genetic counselor sessions for individual patients. So with that, I will pass it back to you, Matt, to finish up.
Thanks, Ken. I hope what we did today was really discuss an overview of how imaging has advanced recently to provide support for gene therapy research. We've attempted to identify some of the main challenges and questions in this space and how imaging assays add value. We really spent the bulk of the time talking about pharmacokinetics, understanding the distribution, whole body distribution of the agent rather than simply relying on the collection of individual tissues. However, we also showed that the collection of individual tissues is critical to the full assessment. We believe that the whole body imaging allows you to hone in on the time frame in which to collect those tissues, and also gives you an opportunity to look at both target and non-target tissues. In many cases, the non-target tissues can be very difficult to anticipate or to guess before the study is even conducted. Understanding expression and downstream dynamics, whether or not there's a response or an efficacy biomarker, such as flumazenil, that may be directly related to the drug effect is critical. What we didn't review is the host of other clinical imaging tools that are deployed routinely to evaluate disease and effect on therapy. But certainly there are many other tools used daily in the clinic and in research that are used as PD biomarkers. We believe that there's proven value in preclinical research. However, we also believe that these are relatively new, underutilized tools. And we are all still beginning to learn the value that they can provide us. And we're also on the precipice of seeing advanced clinical applications. Some of these tools are being deployed in early clinical studies for the first time. We believe that they're poised to advance gene therapy research to help perhaps with the selection of patients, but certainly as a proof of concept to ensure that you're able to demonstrate distribution of your drug to the target tissue. Certainly one of the larger challenges that pharma faces that we didn't address today is that intermediate clinical result, in which case maybe a couple of patients responded and maybe a couple of patients didn't. And without use of biomarkers, there really is no way to know what really happened. What we've tried to demonstrate today is that there are imaging tools to assess distribution, to confirm that you're able to get to the sites of interest, to evaluate happens in non-target sites, and then also to understand that there is a dynamic effect that is related to your intended hypothesis or your mechanism of action. With that, and on behalf of Ken and others here at Invicra, thank you very much. We're happy to address any questions that you may have.
Thank you, Dr. Bloom and Dr. Silva, for your insights. We are now going to open it up for our Q&A session. As a reminder, attendees, please use the Q&A box for questions so that we may address them accordingly. The first Q&A question we have, I believe, is for Dr. Bloom here. What are the advantages of the PID technology over standard IHC?
Thanks, great question. So we believe that there are many advantages. The biggest advantages, I think, really come down to higher sensitivity detection without an amplification step. So when you have a small amount of protein or small epitopes that you want to be able to detect, one of the problems with routine immunohistochemistry is that the chromogens that we typically use are not sensitive enough to show low level of epitope. So what we do to to visualize that is we enhance the detection system, but we do that through amplification processes, which introduce noise into the process, which creates what I call in immunohistochemistry, the hallucinatory stain, where you see very weak staining, but there's also very weak staining in the background that you've introduced through the amplification process. PIDs, since they're so bright, and since the process does not involve an amplification step, we believe that we can get down into visualizing even a few molecules that might be present on a cell. So high sensitivity for sure. But one of the other issues with chromogenic assays, especially chromogens like DAB, which is routinely used in standard immunohistochemistry, is that its absorption patterns aren't linear and they saturate staining very, very quickly. So it's not a very good stain if you want to quantify the amount of epitope. So it's terrible at the lower end of the spectrum. It saturates out quickly, so it's not good at the high end of the spectrum, and it's not very linear in between. PID solves all of those solutions. So not only does it get you low-level detection, it has great linearity, and the signal does not saturate easily. So you can see that high level of expression, even with strong expression of epitopes.
Thanks, Dr. Bloom. Here's a question, I believe, for you, Dr. Silva. When is the drug development research process the right time to initiate this type of research?
Yeah, it's an excellent question and a somewhat difficult one to answer. The short form of that is as early as possible. However, we've seen this research assist in really two ways. In some cases, it's specifically to evaluate a drug candidate, in which case, we'd like to begin in the ideally in the lead op stage or maybe hit to lead stage of drug discovery, where they've identified, the pharma company has identified some chemical matter or gene product that is at least worth studying. In some cases, we can utilize these tools for screening to help evaluate different delivery systems or to evaluate different constructs and to determine which are more effective than others. So in general, that would be the time. We've also seen value when deployed to support a platform research. So if this is something that the company is doing as a large platform, it is perhaps even a little bit earlier than that. These methodologies can be deployed quite early to set them up for success for future studies. In general, a short answer, if the intention is to take this all the way to the clinic, it generally takes 1 to 2 years of research to get us to the point where we can deploy some of these tools in a clinical setting.
Great, thank you, Dr. Silva. Dr. Bloom, I think this is another one for you. Does the PID method require a change to the standard IHC process?
So the short answer is, in most cases, no. It is a secondary label, so you can apply your standard antigen retrieval methodologies, your standard primary stain, and then the PIDs are attached to a secondary antibody. So it would be attached to an anti-rabbit or an anti-goat, for example. And so should work with, for the most part, with routine immunohistochemistry.
Okay, great. And we have one more question. Can MRI or CT or other non-nuclear methods be used rather than PET or SPECT? Dr. Silva, can you take this one?
Happy to, yeah, and it's a good question. The first example I showed was an MRI example in which, rather than looking at directly at the AAV or the ASO, or we've used these constructs interchangeably, I think, in the course of this talk. They're obviously quite different than one another, but in that case, an MR contrast agent was administered along with the gene therapy product. And while those are good methodologies and there are cell labeling technologies, MRI does suffer from a sensitivity problem that is overcome by nuclear medicine. So in most cases, to effectively evaluate small quantities and decreasing quantities as clearance and distribution take hold. Really, the nuclear methodologies are far superior. That being said, at the pharmacodynamic stage, where you really want to evaluate a response, there are several options by MR or CT that can be deployed there. And that's something we should consider having a webinar on in the future.
Great. Thank you for attending our webinar. Be sure to visit invicro.com or follow us on LinkedIn for future sessions. This concludes today's webinar. Thank you, Dr. Silva and Dr. Bloom, for hosting this.